Did you think ever that this long polypeptide chain will how can compress, or you can say is how can fit into a small cell or small our uh, nucleus of cell or small membrane? So this is the magic that created by the universe, right? But how it is possible? And the answer is that folding property of our building blocks, right? Hey guys, welcome to Creative Biology, and here in this lecture we are going to discuss how is this possible for the protein. Before that, we must have to some prior knowledge about that peptide bond, and for that you need to watch my previous videos, so go and check that. Okay, and now first of all, that you need to understand that three, or we can say the three main functions of any molecules, that is an bond, angle and plane. Now for the better understanding of the peptide bond or its rotation or its secondary structure of the protein, you have to understand the three bonds, that is and omega, phi and psi, and angles, that is again in phi and psi, and planes, that is two plane, that is phi and psi plane in here in case of the protein. Okay, now what is phi, psi and we can say is this omega. So simply that peptide bond is known as an omega here, that due to the lone pair of electron it contained a partial double bond characteristic, right? So that bond is here known as an omega. Then phi bond, that is, and we can say that n and bond between n and alpha carbon, or you can say bond between nitrogen atom and alpha carbon, that is known as an phi bond. That if you draw a simple structure of amino acid, that this 4 valency of carbon here, that is an carboxyl group, that we can say is one hydrogen, one R chain and one amino group, right? So this bond, that is, or this called the alpha carbon, so this bond is phi bond. Now what is psi bond? Psi bond is simply a bond between alpha carbon and your carbonyl carbon. So here in case of this simple structure, if I explain, this is the phi bond between the alpha carbon and your carbonyl carbon. Okay, now what is angle, or we can say how angle form? That angle is formed between the two bond, as you can see if here that this is an angle. So here you need to understand two angle, that is an phi and psi angle. So when this phi bond is generated, or we can say is rotated, that angle creates that known as an phi angle, and in case of sine, that known as an psi angle. Or what is plane? That if we understand this whole mechanism in the 3-dimension view, then we need to understand the mechanism, or we can say is terms, that plane. So here in case of this, that two plane, that is an phi and psi again, that is rotated against this or we can say around this alpha carbon, so that plane is generated like this. So here as you can see, so that two planes, see that one or that one, that is phi plane and that is sine plane. That two plane rotate around this alpha carbon. Why these two bonds are only rotating, but this omega bond will not rotate ever, because of this partial double bond characteristic. It cannot allow to rotate this bond. So in the peptide chain, or we can say in the polypeptide chain, only two bond, that is an phi and psi, rotate around each other and make the secondary structure of the protein. Okay, that is a simple mechanism. So that rotation between these two planes, that is in phi and sin, right, that is responsible for its second structure of the any of the protein. You can take any of the proteins and you find this type of the rotation there. But this rotation is not that much simple as it seems, because of this steric hindrance. Now what is steric hindrance? Steric hindrance, or we can say inhibition or resistance during these rotations, is due to the R side chain here. That is an R side chain, right? So due to this R side chain, we can say, if in this case, that is, this is alpha carbon, right? So it also contain an R chain with an hydrogen molecule. So due to this R chain or complexity of these R chains, that two planes are not simply rotated around each other, because it creates the resistance. Now just imagine that if these two planes are rotating around this carbon, these R chains are there, that due to its complexity it will generate a resistance between this rotation, right? So that is known as an steric hindrance, or we can say steric inhibitions of the planes, that alpha, or sorry, that is phi and psi planes. So that is one thing. And if you know this much terms, you can easily understand the Ramachandran plot. Ramachandran plot is not that much complicated. Okay, now let's take an example of one simple amino acid and one complex amino acid. As in simple amino acid, I am taking the glycine. So as we all know that glycine is the simplest amino acid because it contains only hydrogen as an R chain, and a complex one, that is an proline, because it's contains the cyclic chain instead of this R group, right? So in case of glycines, as we know that glycine doesn't have any complexity in R chain, so in the rotation of this phi and psi plane, that is not any steric hindrance generated. But in the proline, that steric hindrance due to its group, that is R chain, is very much higher. So that is an example. So that's all about this dihedral angle, or we can say is angles, bonds and planes, right? And now let's see the Ramachandran plot. So Ramachandran plot is simply two-dimensional graphical view of this phi and psi planes. So now let's see how it plots. So Ramachandran plot, you just have to consider the two-dimensional graph. As you can see here, this is the two-dimensional graph I plotted here, that is from plus 180 to the minus 180 degree. Okay, that is in two plane, that is an psi plane, that is on the y-axis, and this is the phi plane on the x-axis. Okay, now this when this rotation occurs, that rotations will occur on the basis of some bonds, or we can say is angle created when this plane to plane rotates. So based on that planes, this Ramachandran plot will allow us, or tell us that this polypeptide chain will convert into this type of the secondary structure of the protein, right? So here, if that angle, or we can say angle or degree of phi and psi is between this value, right? So that if that psi value is near that 120 and that phi value is near about minus 120, then that type of peptide chain will create a beta sheet. So here, that if that is an in plus sign, so that called the alpha, and again this is alpha. But difference between these two plane, that is a right side, or we can say is a right graph, that amino acid that consider here will form left-handed alpha call, while in this that called the right-handed alpha turn. Okay, and in this that two region will separated, right? So this one that is in purple in the color will create, or we can say will convert into the secondary structure of a beta plated sheet, and this will also converted into the beta plated sheet. But this is for the anti-parallel beta sheet, and this is for the, that is in purple region, is for the parallel beta sheet. Okay, so that's all about that. Right folder, or we can say right quarter, is for the left-handed alpha helix. In the left quarter, that first is of beta plated sheet, and that second one is of right-handed alpha helix. Okay, there is some software is there for the if you have the some unknown polypeptide chain and you need to know about its folding properties, so there is some software for that. You can easily know about the folding property of that unknown proteins, right? And after knowing its folding property, you can easily know about how it's generate its secondary structure. Okay, so that's all about. If you like this video, please thumbs up and subscribe to our channel. Thank you.